Okay, ready? Ready? Have you guys had a chance to review a little bit? Good. Let's see how you do. These are excellent test questions. You ready? What are the four basic tissue types? Epithelial, CT, muscle, nervous. Excellent. What is epithelia versus glands? Epithelia covers. The secret word covers, or yes, covered like covering your skin, covers surfaces. And then glands? Secretes. Glands secrete. And on that note, what is a gland anyway? What is a gland? Remember? What do we say? Gland is just a group of tissues, group of cells that carry out a certain function, like secreting a substance into the inside or outside. Very good. Yeah? Okay. How about exocrine versus endocrine? Exo, getting rid of outside of the body, a duct leading to the outside of the body, like sweat or oil glands. And endocrine, interstitial. It's the same thing. It's a gland, but instead of a ductway passageway leading to the outside, it leads to the blood inside. It never leaves the gland. The substance never leaves the body. Very good. All right, give me at least 4 characteristics that is true of all epithelial cells. Do you remember? Like looking at one epithelial cell.
I was gonna say they touch each other.
They touch each other. That's true. They physically, literally touch each other. They all have a basement membrane. They all touch a basement membrane. Yes.
They have a.
Yes. Very good. Yes, they have a top. They have direction. What's the top called? Yes.
Something.
Yes. Apical. Very good. And then the bottom. The base, basement membrane, which touches the basement membrane the basal layer,
Ganglia.
Very good. Excellent, you guys. All right. Do you remember the junctions? Yeah, basal lamina, basement membrane. Do you remember the junctions? How many junctions did I say you need to know?
3.
4. There's more, but you only have to know 4. The one that's closest to the apical surface is the tight junctions, or you might find it as the occluding junction. Excellent. Tight junction, what is it like, or what does it do? It seals for
molecules to not go.
Yeah, for like aqueous substances, so that we don't dissolve when we go when we jump in a pool. Keeping water out of the inside of the body or inside of an organ. Very good. That's a tight junction, and it seals like what substance did I tell you? What was the example? It seals like silicone. The next one down, or after that, what would you find? Desmosomes. Desmosomes are the ones that, give me some characteristics. Lock. That lock what to what?
One cell to another.
From one cell to another cell, like sideways. From a neighbor cell to another neighbor cell. Desmosomes. Those are the strength. Okay? Those are like the tearing, that they keep the cells from tearing apart. Now, what are the desmosomes like? What? Silicone or nail, like nails or screws. Yes, exactly. Excellent. All right. Next one? The gap junctions. Those allow what kind of stuff to go through? For communication, like ions, like sodium, potassium, whatever, like ions to go through for the purpose of communication. All right. Lastly, the one that was sitting right on the bottom there, the hemidesmosome. Excellent. The hemidesmosome, like the half of a desmosome, hemidesmosome. And those ones
Back to the basement of the cell. Block basement.
They lock the basement membrane, they lock the cell to the basement membrane. Yes.
I thought it was the adhesion cells.
No, that, we're not gonna do that. We're not gonna do that one. That's this one right here. Yeah, basement membrane. Yeah, there's a bunch more, but those four, I'm happy you guys got those. Very good. Next question. Are you ready? Where might you expect to find simple location? Simple squamous epithelium. What was it? Kidney? Give me, it's possible, give me one that, simple squamous, give me one that, the one that I mentioned in class, simple squamous. What are they good for? What do they look like?
Skin?
Fried egg.
Not skin, simple squamous, just a very thin sheet, fried egg, yeah. All together collectively though, it's a very, very, very thin membrane. Good for?
Lungs. Eyes.
What was the,
Oh,
what
yeah.
was?
Lungs.
Lungs. The lungs was the example I gave you, the lungs. Good for gas.
Oh, diffusion.
Gas diffusion, very good. Make it very easy. Excellent. Give me a location for stratified squamous epithelium. Location, stratified squamous.
So.
The skin, excellent, excellent. Inside of your cheek, gums, outside, excellent. Stratified squamous. Give me a location for simple cuboidal.
Glands.
Intestine, maybe some parts of the intestine. Glands for sure. Glands for sure. Ducts, ductway, passageway, glands. How about stratified cuboidal?
Bladder.
Same idea. Glands, passageways, those have the same locations. Kidneys, stuff like that. Okay. How about simple columnar? Where would you find that?
Small intestine.
Yeah, the intestine. You could say like the GI tract. That's the simple columnar. Okay, now how about stratified columnar? Stratified columnar. This one's kind of rare. It's not that common. Stratified columnar, but you might find it like in some some. What's that? Esophagus? Yeah. There's certain like salivary glands, but it's definitely the most rare out of all of them. Okay, now how about the pseudostratified ciliated columnar epithelium?
Oh. Oh no, I
The,
don't.
the trachea. The trachea. I heard it. The trachea. Remember the mucus escalator? The trachea, because of that cilia producing mucus and sending it up. The trachea, the nasal passageways, that's the pseudostratified ciliated columnar epithelium. Excellent. Okay, another question. Your gums are made specifically out of what? Your gums are made specifically out of what? Very good. That's about as broad as you get. Made out of cells? That's probably. Huh? Okay, what's more general? Okay, cells, tissue, epithelia. It's stratified. It's stratified squamous epithelium. Now, more specific than that. What's the fancy words? I'm looking for two other words. Keratinized and non-keratinized.
I was just looking
Gums?
at that. Oh my god.
Gums are?
Non-keratinized.
Non-keratinized. Doesn't have that special protein keratin. Now, where would you find keratinized, simple, squamous epithelium?
Start with the skin.
The skin. Vagina, anus, anything like that, that would be non-keratinized, and anything that stays dry is keratinized. Excellent. Very good, you guys. All right, now tell me everything you know about transitional epithelium. Give me everything you know. Transitional. You find it in the urinary bladder because it transitions very easily, all the time, right?
Oils.
Stretched out when you're fully full, when your bladder is full, it totally stretched out, it like flattens, and then after you urinate, the bladder comes together, and then it looks more like what you see up on the screen in the slides. What else? So we said location. We know that it's what it's good for or why it does that. It recoils very easily. Excellent. All right, and histologically speaking, it's the one that's kind of like, it's really hard to differentiate. It's just sort of a bunch of like water droplets going every which way. Okay, how many different kinds of exocrine glands are there? How many different kinds?
Oh, very good.
2, 3. Remember the 3? Remember we were talking about if we divided them according to how they release substance. What are their names? 1, 2, 3. Remember the first one we talked about starts with M? Merocrine. The second one, merocrine, exocrine. The second one was? Which one? Apocrine. And third?
Endocrine.
Holocrine. So what is it about, what's so fancy about the merocrine? What is it? Tell me what you know. What do you know?
Merocrine secretes.
Merocrine secretes by exocytosis. Remember, going through the ER and the Golgi by exocytosis. Uses little vesicles to transport, like in Gen Bio. Now, how about apocrine? Yes, it's the name. Apical, apocrine, just based on the name. It slices the top half of the cell, not the entire cell, but the top half of the cell actually cleaves and releases its substance. And finally, holocrine, it's in the name. It bursts. It bursts. The entire thing explodes, hence the word holocrine, whole, the entire cell. Excellent. Okay, moving on. Tell me everything you know about in general, connective tissue. Connective tissue, what were some of the characteristics we said? By far the most, the most broad, because we're talking there's blood and there's fat and blood and supporting. Okay, by far the most broad. That's what makes it like the hardest. Okay, if you were to divide CT into 3 different categories, subcategories, what would they be? Remember the first one? CT proper. Excellent. CT proper. Number 2, fluid CT, and lastly, supporting connective tissue. Very good. I think that's all. Maybe one more question here. What do the 3 different types of fibers look like under the microscope? Remember? First off, what are the 3? Name them. Collagen and reticular and elastic fibers. 3 different types of fibers. Collagen look like?
Broad.
Yes, pink, light pink, broad bands. Very good. And then reticular look like? Olaf's hands. Olaf's hands, like little branches. And then finally, elastic look like
Stretchy. Straight.
kind of string. Very, what's the word? Very dark, high contrast. Very, very, very characteristic. The 3 are like completely different, and they're long. They're long. I mean, I know reticular are kind of dark, but they're also very, very short and kind of branchy compared to long, stringy type of elastic. All right, this might be the last question. What are the 3 different types of loose? This is kind of like where we stopped. What are the 3 different types of loose connective tissue? Number 1, which? Areolar CT. Number 2, adipose CT. And number 3, reticular CT. Excellent. Okay, let's get out of here. And let's go back to where we left off. And we start where we left off, which is okay, right here. Now, what was that mnemonic device? You remember? Yeah, PARD, P-A-R-D. P is the top of the dermis, is the top. I know it's not, if it's not that clear, or if you just, don't worry about it, we're gonna get to it next week. But in the dermis, you have the epidermis, the dermis, and the hypodermis of the skin. Epidermis, dermis, hypodermis. Of the dermis, you can split it up into 1/5 and 4/5. The top 1/5 we call the papillary layer, because it's the part of the layer that look like fingers. And PA, so papillary layer, contains A, which is areolar tissue. Now, the PAR, which is the 4/5, that's the reticular layer, and the reticular layer contains the D, which is dense, irregular connective tissue. Excellent. All right. Where do we find adipose tissue? Yeah, well, it's made out. Yeah, it's fat. So where do you find fat? Two places in particular. The stomach. Yeah. Well, remember, the adipose tissue found in the skin is not found in the skin, because, as we'll see next week, technically speaking, skin is only the epidermis and the dermis, but it is found in the hypodermis. It is found in the hypodermis. Okay, so it's dominated. Adipose tissue dominates the hypodermis, or you can say the hypodermis is dominated by adipocytes. You could also find it surrounding viscera, all your organs. So the subcutaneous, that's the stuff that's not as bad for you. You know, you have like these, skinny little dudes, and they look healthy as ever, and they're dying, so they die so early, versus a more, big boned person, and they both go to the doctor, and they find that the larger big boned person is significantly, by all standards, significantly healthier, because they'll have less of the visceral fat, even though they may have more of the subcutaneous fat. It's the visceral fat that kills you, the fat like around the heart, for example. Subcutaneous fat is not nearly as dangerous. Here's what it looks like. Squashed, sort of like simple squamous, except the cells are so fat they push the nucleus off to the corner. All right, and lastly, the reticular tissue. This stuff is found, we generally show it like in liver. If you can remember these three locations, I would be happy. In the kidney, in the liver, and the spleen. And here's the function. It provides support and resists distortion. The word itself means like a network, forming like a network, providing support and resist distortion. That's reticular tissue. Remember, keep them straight, because there's reticular fibers, little branches, looks like little Olaf hands, that's inside of the reticular tissue. All right, now, just like there were 3 types of loose connective tissue, there are 3 types of dense connective tissue. Just like there are 3 types of loose, there's 3 types of dense. The first one is dense regular CT. The second is dense irregular, and the third is elastic. Okay. these should be, the dense regular should be fairly easy to remember, because if they're dense, very compact, and regular, which means the bands are very, very what? Very, very parallel to each other, stacking very easily. Well, that type of tissue can take stress very well from two directions, like you would want in tendons and ligaments. At the ends of your bones, at the ends of your muscles. Muscles pull on bones, at least skeletal muscle pull on bones. And so you'd want to have that. Dense regular. Now, dense irregular is not as strong as dense regular, because instead of taking stress from just two directions, it takes stress from several directions, and that's why the fibers look like they're going in every direction, because it can take stress from all kinds of direction, versus up here, dense regular, taking stress more or less from just two directions. Does that make sense? Where would you find dense irregular? We find this covering the visceral organs, in the superficial layers of bones and cartilage, and even in the dermis. We'll find where specifically? Do you guys remember? Where in the dermis? Remember? The lower, the reticular layer, the lower 4/5. There it is again. That's what this was. That was the D in the PARD, the lower 4/5 of the dermis. Dense irregular. And once we get to it, once we get to this, to the integument system, next week, we'll see it's very, very, very clear, very obvious. You can't miss it. You just have to sort of look out for it. No consistent pattern of fiber, that's your dense. And finally, in elastic tissue, the third of the dense CTs, there's significantly more elastic fibers, very, very, very characteristic, a lot more than the collagen. It looks like this, it's a lot of elastic. And so it's very, very springy, resilient. Elastic tissue is found between vertebrae in the walls of large blood vessels, like arteries, and in the erectile tissues of the penis. This type of tissue, elastic tissue, needs to be able to be stretched and recoiled very, very easily. Definitely, we can see that in arteries. Elastic tissue, not elastic cartilage, elastic tissue. Elastic tissue, elastic cartilage, elastic fibers. 3 different kinds of things. In this example, we have elastic tissue made out of lots and lots of elastic fibers. Okay, we're going to go through this really, really quickly, because this will be this is 2 entire units in, 2 entire chapters in A&P2. So this is all basically that we're going to say. All I'll really expect you to know, since we're going to go on this in depth, is the blood of the fluid CT transports nutrients to cells and rids the cells of the waste by transport, and the lymph is in close, works in close with the immune system by filtering the blood. Lymph is, the nature of lymph is very, very close to the composition of blood. It's basically blood just missing a few things. And so lymph is filtered and is dumped back into the blood system. Okay, now cartilage. we break this up, chondrocytes, chondro meaning cartilage, cyte meaning cell. These cells are sit inside of these lacunae or lacuna, small little homes. And so if we are to divide these three guys into these three types of cartilage, you got hyaline cartilage, you have elastic cartilage, and finally you have fibrocartilage, or you can say fibrous cartilage. Just like in lab, you look at these, what are the differences? Just by looking at these histology slides, these cells are very plump. The lacuna are very plump, whereas the fibrocartilage lacuna and cell are very are much more elongated or elliptical. Again, the elastic is very purple, and then another big difference between the fibrocartilage and the hyaline cartilage, the fibrocartilage has a lot of swirls. You can even see it in the cartoon picture, swirly. Whereas in the hyaline cartilage, it's more like a watercolor, there's no real swirls. All right, location, hyaline cartilage, you wave your hand, hyaline cartilage. Hyaline cartilage is really, it's found at the ends of your long bones. In the legs, location of elastic cartilage, like in the ear, very elasticy. And finally, fibrocartilage is found, one location is found in the intervertebral discs, between your vertebrae. Now, what would be the use, or what is the function then of these three cartilages? Hyaline cartilage needs to be extremely rigid, almost the same consistency, the same hardness as bone, because it bears a lot of weight, especially in the knees and the ankles and in the leg bones, bearing a lot of weight. Very, very rigid. I'll use that word rigid on the test. Imagine if you had elastic cartilage where you should have hyaline cartilage. Elastic cartilage at the ends of your bones. This stuff would squish, and your ends of your bones would grind against each other. It would be extremely painful. You need something kind of tough. This is the cartilage that you bite off at B-dubs, eating your chicken wings. Hyaline cartilage. Elastic cartilage, very elasticy, and fibrocartilage is in the middle. Elastic cartilage is the least rigid, but fibrocartilage is somewhere in between as far as rigidity. It's not quite as hard as hyaline cartilage. It's not quite as rigid as hyaline cartilage, and it's not quite, but it's significantly more rigid than elastic. You got to have a little bit of give and take, for example, in the intervertebral discs, because they have to bear some weight. For example, that the cervical vertebrae have to hold the head, and the thoracic vertebrae have to hold the head and the cervical vertebrae, and the lumbar vertebrae have to hold the head and the cervical and the thoracic, so they hold weight. But they also have to have some sort of elasticity, because you need to be able to move back and forth. That's what allows you, that lateral flexion back and forth is allowed for by this elasticity of the fibrocartilage. So it can't be extremely elastic, it can't be extremely rigid, because then you wouldn't have any motion. It can't be too elasticy, otherwise those bones would grind against each other. You gotta have something in the middle. And so this is medium as far as this fibrocartilage is medium as far as rigidity. Look for that word on the exam, rigidity. I don't like to use the word hard, because you might say hyaline cartilage is very hard, but it's also very brittle. So, there's all these terms are sort of relative, how we use them. So I'll use the word rigid. Very, very rigid. The least of the rigid. Elastic cartilage is the least rigid of the three, and then fibrocartilage is somewhere in the middle. Okay. Osseous tissue. Osseous tissue. Remember, on the practical, you'll get only half credit if you say bone. You gotta say compact bone, compact bone. Well, there's two types. There's compact bone, which is on the outer surface, and you have spongy bone lining the internal cavity, and that's all we'll say about this particular slide because we have an entire unit for just bone. But a little bit of review as far as the histology. This one unit, this one entire unit right here, this one unit here is called an osteon. You gotta know that for lab, osteon. Inside the osteon, you have these osteocytes, which are mature bone cells, which laid out these layers. The layers, 1, 2, 3, 4, and so on, in this one osteon are called concentric lamellae. Concentric meaning going in a circle. This middle canal is called the central canal. And finally, there's these little canals, these little rivers going perpendicular to the layers. And those layers and those little rivers are called canaliculi. Canaliculi meaning little canals, connecting from cell to cell, connecting the blood passageways filled with blood. Four types of mucous membranes. Number one, mucous membranes. These guys line organs that communicate to the exterior. Mucus, they must be kept moist to facilitate movement and absorption or secretion. You have serous membranes, and once again, we've seen these three words now, these three cavities, the pleural cavity, the peritoneal cavity, and the pericardial cavity. And what were the two words that are always put in front of those three words? Remember, if we're talking up here, pleural is lungs, pleural is lungs, so we either use, and there's a whole bunch of P's here. There was that visceral or parietal. Visceral or parietal. So parietal starts with a P, pleural starts with a P, peritoneum starts with a P, pericardium starts with a P. A whole bunch of P's to get you completely confused. Well, in front of these three words, you'll either say parietal or visceral. There's this protective bag that protects your viscera from the rest of the body cavity, from the lining of the body cavity. A bag. And half of that lining, the stuff that's touching this side, is called parietal. And if it's up here in the lungs, where the lungs would be, then it's called parietal pleural. Parietal pleura. And as it goes around and touches the lungs, so it'll do this, and then it'll touch the lungs, that lining then would not be called parietal pleura anymore, but it would be called visceral pleura. Parietal pleura, and then as it goes around, visceral pleura. Visceral for touching any kind of viscera. The same idea, peritoneum, below the diaphragm. Below the diaphragm. As you cut your fetal pigs, you're going to have to sort of pull and snip, and what you'll be holding on the outside here is called parietal peritoneum. Parietal peritoneum on the outside, and anything that's touching the actual viscera will be called visceral peritoneum. Same thing with the pericardium. So there's those three words for you. Just describing the different cavities, the lining of the cavities. And number three, the cutaneous membrane covering the skin, relatively thick, dry, comparatively speaking, relatively cutaneous membrane. And finally, the synovial membranes. Most of our joints are synovial joints. The ones that move really, really freely like in our fingers and our toes, our arms, our legs, our knees. These are all synovial joints. And each two bones that articulate meet up in what's called a synovial joint cavity, filled with synovial fluid secreted by the synovial membrane. Synovial, synovial, synovial. So there is the fourth type of membrane called synovial membrane. And by the way, the synovial fluid, which fills up the joint cavities, is what brings in the nutrients to the joint and gets rid of nutrients away from the joints, since joints are, once again, avascular, because cartilage is avascular. There's no other way to get nutrients in or out, since there's no blood supply. All right. By mass, muscle tissue makes up 50% of the body. By mass, 50%. Now, that's quite impressive, since connective tissue consists of, you're supporting your adipose tissue and your tendons and your ligaments and your blood. Well, muscle tissue is just muscle tissue. That's pretty impressive. There's three types. Skeletal muscle pulls on bones, allows for joint movement, allows locomotion, for locomotion. Skeletal muscle. Cardiac muscle pumps blood through the body, and smooth muscle moves food or materials through the body. Smooth muscle lines vessels like arteries and veins, and when the smooth muscle contracts, vasocontracts, the artery constricts, and so it's we don't only, there's more function than just what's here. These are just a few of them. Skeletal motion, and there's also one thing to take note of. This is also very important. Our body stays at 98.6 degrees Fahrenheit because of the waste that's produced by the movement of skeletal muscle. That waste is not really waste. We use it to keep our bodies warm, which is why when we lay still at night, we need a blanket, because we stop moving. We're not generating heat from motion because we're not moving. Hence, we need a blanket to keep warm. Skeletal muscle is multinucleated. It's also striated, just like cardiac muscle. Cardiac muscle also has intercalated discs, and smooth muscle is called smooth because it's not striated, and they're also single. They only have one nucleus per cell. They're called smooth because they look smooth, because they don't have that zebra print, those striations. And one more thing to take note of, since these might look the same, although cardiac generally looks purple. When it's stained, it looks purple, versus this looks the reddest, and this generally looks pretty faint. Cardiac is also branched. Cardiac muscle is branched, so the heart can contract all at the same time. Two types of nerve cells: neurons and neuroglia. Neurons and neuroglia, or you could say glial cells. The neuron, keep in mind, there's a difference between a neuron and a nerve. A neuron is one cell. A nerve is a bundle of cells. Neurons are the long ones. Those are the guys that the shortcut hand look like this. Here's the shortcut hand. You got the cell body, the axon that looks like this. This is the shorthand. And when you zoom in, they got these dendrites that look like this. And this is the cell body here. Here's the cell body. And then you got your axon like that. Those are the three parts. That's all you got to know for this unit, since we have an entire nervous unit that we're going to go through. Where do you find neurons? In the central nervous system and in the PNS. All throughout the arms, the legs, that's where you find neurons. Where do you find the other type of cell, the neuroglia? And there's several types of neuroglia. These guys you find in the brain. What do these guys function? These are the supporting cells. They repair, for example, they maintain physical structure, they provide nutrients, but all the neurons do, these guys are only communicate. They're only for communication, sending a signal to another cell, to another neuron, from neuron to neuron, or neuron to muscle, or something like that. Communication. And that concludes the histology chapter.